Very good afternoon to one and all. I, Karu Assistant Professor, Department of Pharmaceutics, RBVRR Women's College of Pharmacy, welcome you all to the virtual guest lecture on the topic Pharmacovigillance in Drug Discovery. I now invite our Principal Ma'am, Professor M. Sumakant Garu, to address the gathering. I welcome you, Ma'am.
Thank you, Anushree. It's my honor to welcome today's eminent personality, Dr. Rao Vadlamudi, and it's my privilege to invite all the participants, students, and colleagues, and the faculty members. A very good afternoon to one and all. Yeah, today we are conducting this guest lecture on pharmacovigilance and drug discovery. The aim of this lecture is to aware all the pharma professionals what is pharmacovigilance, how extent it is useful in drug discovery, and how it is helping in practice school for the students who are in final year in B pharmacy. So as per PCA curriculum, the students have to select this one of the elective subjects which were mentioned in semester 8. With this lecture, the student can able to understand how he can or she can do this project in pharmacovigilance as one of the elective subjects as per the PCA curriculum. So the main intention is to aware especially the final year students about their practice school to take up the project on this pharmacovigilance. So with this brief, I am here inviting Professor Rao, Dr. Rao Vadlamudi sir, please. Yeah. Now I request Shailaja, Dr. Shailaja, to introduce sir. Over to Shailaja.
Thank you. It's my privilege and honor to introduce today's speaker, Professor Dr. Vadlamudi sir, who is working as President Commonwealth Pharmacist Association. Sir has completed his B.Pharm and M.Pharm from Visakhapatnam, India. He obtained his PhD in Pharmacology and Toxicology Department in 1983 from the University of British Columbia, Vancouver, Canada. Then he started his career in 1984 at UDCT, Mumbai. During his career of 35 years, he held so many positions in academic as well as in industry. Coming to his academic positions, Dr. Vadlamudi sir worked as a reader in Pharmacology Department, UDCT. Later, he promoted as professor in pharmacology in Bombay College of Pharmacy. Then he worked as a director of BCP and as well as he continued as a director in St. Peter's Institute of Pharmaceutical Sciences, Warangal. Sir worked as adjunct professor in Acharya as well as BM Reddy College of Pharmacy, Bangalore. Coming to his industrial career, he started working with HS Research Center as a head pharmacology department in 1990. Then he was promoted as a head pharmacology department division in the Peramal Research Center. Later, he worked as a vice president in Suven Life Sciences and he worked as a principal fellow at Nectar Therapeutics India Limited. Then he worked as a senior vice president at Vimta Labs, Hyderabad. Coming to his awards and rewards, he worked as an editor of Indian Journal of Pharmaceutical Sciences, the official scientific publication of Indian Pharmaceutical Association in the period 1996 to 2014. Then he served as the president of Indian Pharmaceutical Association for 2 terms, from 2014 to 16 and 2016 to 18. During this period, he acted as a member of Drug Technical Advisory Board of the Ministry of Health and Family Welfare, Government of India. Currently, Sir is working as a president of Commonwealth Pharmacist Association. He has re-elected for the term 2020 to 2022 and he is a member of governing body of Life Sciences Sector Skill Development Council of India. He is also the professional secretary of a forum of all national pharmaceutical associations of Southeast in East Asia region countries. He received the fellowship of Indian Pharmaceutical Association in 2000. He received Dr. B.N. Ghosh Memorial Oration Award of Indian Pharmacological Society in 2005. He received Professor M.L. Kharona Memorial Lecture Award of IPA in 2009. Sir received Eminent Pharmacist Award of IPA in 2030, IPA President Special Oration Award in 2014, the Fellowship of Association of Biotechnology and Pharmacy, and the Acharya P.C. Rai Gold Medal Award in November 2019. With this introduction, I cordially invite Dr. Vadlamudi sir to deliver his talk. Thank you, sir. I cordially welcome you, sir. Please deliver your talk in pharmacological vigilance in drug discovery. Hearty welcome you, sir.
Thank you. I hope I'm audible to all the people as well as I'm visible.
Yes.
Yeah, yes.
Yeah, I would also be sharing the screen to show you my presentation.
Yes, sir.
Okay, share screen. I'm not sure whether my screen is being shared. Is it there?
Not yet, sir.
How about now?
No.
So there is usually there would be an option to share screen.
Yes, sir.
Share. Application window. Yes. Share. How about now?
Yes, sir.
How about now?
Yes, sir.
Okay. But my window, my face is not being shown on my computer. Usually in one corner it comes. But that's okay. If people are able to see the screen. Today's topic, and thank you very much. First of all, thank you very much for the college RBVRR, Women's College of Pharmacy, and the principal, Dr. Sumakanta, for inviting me to give this talk. And it's an honor to be present here and share some thoughts on pharmacovigilance in drug discovery. That's the topic. Nevertheless, I wanted to say that drug discovery and pharmacovigilance are not connected with each other. It is in continuation of each other. Drug discovery starts and pharmacovigilance starts after the drug is approved. What I would try to achieve during this talk of, Madam, how long do I have? Is that an hour or? Hello?
It's sir, 1 hour, sir, as per your convenience.
That means I can go on for days. So then
anyhow,
I shouldn't be saying this because all the people who are watching on YouTube streaming would just immediately switch off and go away. Anyway,
okay.
I would try to complete that in 45 to 50 minutes or maybe 1 hour. It depends
Okay,
upon how deeply I get into, because the topics are very, very intense and extensive.
Yeah, it's true.
What I wanted to convey to you is that drug discovery and approval is a process, and once a drug is discovered, developed, and approved by a regulatory authority, the actual pharmacovigilance on that drug starts. Now, in another way to explain this is that a drug during phases of discovery and development is not referred to as a drug, but it is referred to as a candidate drug. Candidate drug pending approval. Once it is approved, then it is a clinically useful drug, then it becomes a therapeutic agent. Pharmacologically, the definition of drug is very broad because any exogenous chemical molecule that has an effect on a physiological system. That means when you administer an exogenous chemical and if it is capable of producing a change, modulate the physiological function and produce a discernible or observable change, then that molecule is called the drug, and the process of producing that change is referred to as pharmacology. Among several agents that can produce an effect on a physiological system, all of them are not useful to treat a clinical condition. So all the drugs cannot be therapeutic agents. Nevertheless, some of the drugs, when they produce a change in the physiological system, the change can be detrimental to the physiological system, leading to pathology or cell death or some adverse event, and those effects are referred to as toxic effects. So pharmacological effects broadly cover those effects that can be useful to treat a condition, a disease condition, to produce a therapeutic effect, and those effects that are toxic and detrimental and sometimes can even prevent the therapeutic use of a drug. So pharmacology encompasses both therapeutic effects as well as toxic effects. So while discovering a drug, we are really looking at how the effect of a candidate drug can be useful in the clinic to treat a disease and not produce toxicity. Okay, so the definition of the drug discovery and development part, particularly the objective, is to produce a molecule that can be very rapidly developed into a safe and effective treatment for an unmet medical need. Now, there are two components here. One is the molecule should be effective and can be used as a treatment for an unmet medical need, means a disease condition in the clinic that has no proper treatment. That is an unmet medical need. So we try to discover a drug to produce an effect which is beneficial to that condition in the clinic and produce treatment, efficacious treatment. But at the same time, it should not produce toxicity or it should not be detrimental. So there are two components during drug discovery that we evaluate: safety and efficacy. Now, once a drug is approved after clinical trials, during the clinical trials, there is sufficient investigation into the safety of the molecule. So when a regulator is convinced that the molecule is safe for use in patients, simultaneously the molecule is producing sufficient efficacy to produce a therapeutic outcome, then they will approve the drug. Okay. Once the drug is in the clinic and it is being administered to scores of patients, there should be a monitoring of the safety of that drug continuously while more and more number of patients all over the world start receiving this drug. And that monitoring of safety throughout the life period of a drug is referred to as pharmacovigilance. So this is, I'm trying to connect both the terms, drug discovery and pharmacovigilance. Okay, there is sufficient amount of pharmacovigilance or precautions or investigations into whether the molecule will be safe for clinical use or not, which we normally call as safety evaluation of a candidate drug molecule. So just going back one slide, what I would like to cover today is that different phases of drug discovery and development. Why I'm going to cover those things, in addition to pharmacovigilance, is to demonstrate to you that during the various phases of discovery and development, there is always a concern about the safety of the molecule. So the objective always is it going to be a drug and is it going to be safe? That question always arises. And all the experiments planned during the discovery and development stages has one component of, can we predict the safety of this molecule when it is applied in the clinic? In a manner, in a parallel to the other objective is, can we predict its therapeutic effect or therapeutic efficacy in the clinic while developing the molecule? So we have various stages like lead generation of the drug molecule or a candidate drug, then optimize the candidate drug, try to see that it is drug-like and it produces sufficient efficacy in animal models, and then a preclinical development, a phase in which you generate data to convince a regulator that the molecule actually can produce beneficial effects in the clinic, as evidenced by the studies in experimental or laboratory animals. And the molecule can be developed into a drug formulation, as again evidenced by the development of a drug formulation for that molecule, and that it has sufficient adequate pharmacokinetics, means giving giving again a glance into what kind of drug blood levels are reached and how the drug candidate is handled by, how that would be handled by a volunteer or a patient who receives during clinical trials, and that there is sufficient evidence that it can be given a permission to test in the human. And that stage of preclinical development produces what we call as IND enabling, investigational new drug application enabling data, so that to support and give proper evidence for a regulator to give a permission to test in the human for the first time based on the data that the company submits. And then the clinical development starts, which goes in 3 phases. It can be 4, 5 phases also, like phase 1, phase 2, 2A, 2B, 3, and in phase 3 studies can be large phase 3 studies, which can be 1 study, 2 studies, or 3 studies, how many number of studies that are necessary to produce enough evidence that the candidate drug is effective in the clinic and safe, that it can be given a permission of a new drug approval, that is NDA. And the approval leads to pharmacovigilance. Now I'll quickly go through what goes on during these stages. And in the lead generation, usually the current models applied, current approaches are based on target identification. You have to identify a target which is involved in pathophysiology of a disease. We are not talking about any disease or any target. We're just generally talking about if you are trying to find a drug for a particular disease, then you look into pathophysiology of the disease, and you also look at what could be the important events, important involvement of biological pathways or processes that lead to the pathogenesis. So can we identify points of intervention in those processes? And those points of interventions are referred to as targets. And if we identify a target that could be involved, target is just like a molecule. A target, from my perspective, is a biological molecule. It could be a biological macromolecule, it could be an enzyme, it could be a receptor, it could be an ion channel, or it could be a gene by itself, or it could be a, a regulator, a regulatory molecule that regulates gene expression, anything. There can be several number of targets. And these targets, when they start behaving in a funny manner, that means not appropriate manner, then they change the course of physiology into pathophysiology, and the outcome of that pathophysiological process would result in symptomatology that can be identified as symptoms of a particular disease. This is how we keep on describing the involvement of target. And you validate the target. The process of validation really means that if I identified a target, that means if I identified a gene or a gene product or a protein or an enzyme responsible for development of a disease, if I simply say that it is responsible for the disease is not adequate, can I really modulate that gene or that target under physiological circumstances? But I cannot do that in the human. So I try to use either a cell line or a laboratory animal and try to manipulate the target in a way that it starts to behave exactly in the similar manner that I predict that it behaves in a to produce pathophysiology of a disease. And if I try to mimic the same process in an animal model, and if the target is really involved in pathogenesis of a disease in the animal model, if I manipulate the target, the animal should start producing symptomatology similar to that, or that can be associated with a clinical condition. That means we also develop an animal model of disease by manipulating a target that is probably involved in the pathogenesis of the disease. And once if we succeed in doing that, then we say that the target is now validated, because I think that this enzyme is responsible for a change that would lead to a disease. And if I modulate that enzyme in an animal, the same changes can be seen, and then the animal starts behaving and exhibiting symptoms that are similar to that we observe in the clinic in a patient. This is what I want to simply say. And such a target molecule, if we identify and validate it and establish its association with the disease, then we develop a screening method around the target such that we can screen a number of compounds, possible compounds, chemical compounds or natural products or whatever, on the target or on the biochemical or molecular biology or a cell biology assay that is based on the target, to see whether these molecules can produce a change which is similar to that what is expected that a drug would produce in the pathophysiology in the clinic. That means if let's say, for example, if I say that I have an enzyme and overexpression of that enzyme would lead to a change in the body that would produce an increase in blood pressure. I'm just very simplifying the same entire process. So I develop a screening, a screening assay in which the enzyme is involved, and I try to identify those molecules that would inhibit the enzyme activity. So I'm really searching for enzyme inhibitors, because an enzyme inhibitor, when administered to the clinical in the clinic to the patient, the enzyme activity would be stopped by this particular molecule. And since the overactivity of the enzyme is responsible for the production of hypertension or increase in blood pressure, if you stop the enzyme activity, the hypertension would come down to normal level. It's a very simple extrapolation of what exactly is expected to happen. So the assay development involves enzyme activity, and I test many compounds to see if the enzyme activity can be inhibited. But if the target is a transporter, then I'm looking at what the transporter actually transports across the membrane. If it is a a gene regulator, then I'm looking at, can I express or inhibit gene expression? So based on that, the assays keep changing. However, we need these assays to be developed in a very rapid way. We want these assays to function in a very aggressive manner, that many number of samples can be screened. A process in which, if an assay can be used to screen thousands and thousands of samples in a short time, then these assays are called high throughput screening. So preferably we would convert an assay and then make it high throughput and start screening samples like synthetic samples and in libraries, new molecules synthesized, or existing chemical molecules, or natural products. Or if we can even understand the three-dimensional structure of that particular target, which can be obtained from a X-ray crystal, a crystallized target, and its X-ray crystallographic studies can give us a three-dimensional view of how the target would be. And if we have a substrate for that target, or a ligand for that target is bound to the target, then in the three-dimensional picture we can actually see where the target, where the ligand or the substrate molecule is bound to the target protein, and we can remove that molecule to see exactly the area, the topography of the binding site. Some people call it binding cleft, and try to synthesize molecules to fit that on the computer screen. And these are computer-aided drug design processes. Or we can screen natural product extracts and see which one is active, and then further look at the natural products to see, isolate to see which molecule is responsible. It's a whole iterative process, and highly complicated. But if you understand the entire process, then it becomes quite simple. This is a a cartoon in which the lead generation is explained. So you in one box here it is target, okay, and you can get target molecules, structure, etc., identified, isolated from genomics and proteomics. So you know which gene is responsible for the target. You identify the gene, you clone it, and then you express that. You get large quantities of the target, and evaluate the protein structure, and then take a crystal structure of that through extracrystallographic studies. That would help you in molecular drug design, and you can synthesize molecules on the crystal structure, or you can virtually screen chemical libraries that are available on big databases. This part of the entire process is all computer aided, and it is all done on the screen. But if you validate the target in the laboratory and generate a screening assay and start screening chemical libraries, natural product extracts, combinatorial chemistry libraries, or any molecule mixers or pure molecules on that, then these are really, it's empirical. That means you actually do these experiments in the laboratory, and whatever the computer aided design suggests, these molecules can also be synthesized. That means a designed molecule ultimately has to be synthesized in the laboratory, and then use that in the screening assay to see if it is active. And out of all these processes, if you find active molecules through high throughput screening, and those active molecules are hits. Hit is a molecule that is active on the target. If the target is an enzyme, then an enzyme inhibitor is a hit. If the target is a receptor, then a molecule that can bind with high affinity to that receptor is a hit. However, for a receptor, a binding can lead to either activation of the receptor or silencing the receptor, what we call as agonist and antagonist. So this is all so many things that you need to clarify subsequently to see whether the hit, that means a molecule which can bind to the target, can actually produce effects expected of a drug. And that conversion or that evaluation of hits into lead molecules and the leads to development candidates is what is called lead optimization, the next stage. And in this process, if a hit cannot be generated, cannot be developed into a development candidate, it should fail. And you should eliminate that very rapidly. Only those molecules that can become development candidates, you have to speed up. So lead optimization has an objective, that is how quickly I can convert hits to leads and to development candidates. But then the question comes, what is a lead and what is a development candidate? Okay, now leads or those molecules, they have efficacy. That means these molecules, when you test them in animal models of disease or even in the laboratory, in the in vitro screening assays, they should have high affinity binding or produce activity in low concentrations, which that effect or that activity is actually representative of the clinical efficacy that you are going to see in the clinic. Okay, but however, a molecule that is active should also get absorbed, should also reach the target when you administer to a whole organism or a human. So for a molecule to produce activity, there are two components. One is inherent efficacy, that is ability to bind to the target, and the second is inherent drug likeness. That means the molecule should reach the target. So we have to evaluate both these simultaneously, right? And while evaluating these two, then we would be evaluating the so-called DMPK parameters, so the drug, distribution, metabolism, absorption, distribution, metabolism, ADME or DMPK, drug metabolism and pharmacokinetics, that is one component, or ADME, absorption, distribution, metabolism and elimination. While doing all those things, if the molecules are not possessing those characteristics, then you optimize them. That means you go back and you tinker with the molecule, you synthesize several analogs with an intention that each analog, there is an incremental increase in its efficacy, that means its ability to bind to the target, or incremental increases in drug likeness. That means its absorption characteristics, its drug metabolism or distribution characteristics improve, that when you administer these molecules, they get absorbed and remain in circulation for longer periods, which is ideal for the treatment, and then ultimately get eliminated. While we are doing all these things, you please concentrate on these gold letters here: safety and lack of toxicity. So for a molecule to be a good lead and to be developed into a candidate drug, to pass it through preclinical development, one of the characteristics that is necessary is safety and lack of toxicity. So in the journey of a molecule to become a drug, that means during drug discovery and development, the first parameter that is related to pharmacovigilance is that evaluation of safety and lack of toxicity. So we have, during lead optimization, when we are evaluating molecules for the next stage development or progressing into next stage development, we ask 3 or 4 fundamental questions. Is it good? Efficacy, that means does it have enough binding to the target? What is the affinity? That's the word we use, is that. Is it good affinity? If yes, what is its ability? What is its drug likeness? What are its ADME parameters? Can this be absorbed orally? Or does it have great metabolism, or is it not metabolized easily? Or what enzymes are responsible for its metabolism? While getting metabolized, is the molecule inhibiting the enzyme, or is it competing with other substrates? All these questions are asked. And is it distributed? Is it protein bound? Is it eliminated? What is the route of elimination? Is it eliminated as a conjugate through the kidney, or is it eliminated as such through glomerular filtration, or is it eliminated through other body fluids? All these questions are asked. Then these are the 2 questions, fundamental questions, which are more pharmacodynamic and pharmacokinetic. And then we also ask a question: does it have obvious toxicity? Is it safe? If it has any obvious toxicity at this particular point, a red flag is raised and the molecule is dropped. Safety evaluation would come a little later, because what we consider at this stage is that if the molecule has no toxicity, then it is safe. Okay? Because if it has, great toxicity, that means very obvious toxicity, then it cannot be developed further. So the molecule would be dropped. So, just one second. I have something on the screen that is okay. And then we do efficacy and pharmacokinetics in vivo. So, because most of the evaluations on structure activity, structure property, in vitro evaluation, all are done in vitro on cell lines or in biochemical assays or test tube assays. And ultimately, you start giving it to in vivo animals to see if there is sufficient efficacy and pharmacokinetics. And then the molecule is selected for further development. Okay, so the same thing in a schematic way, it is shown as a primary screening would give hits, and those hits with, I would say, appropriate ADME characteristics, appropriate efficacy, appropriate safety, and lack of toxicity. All these would lead to identifying molecules which are called leads. And the leads are further developed, because at this stage, for the lead development, you need whether the lead can be converted into a dosage form. So we do pre-formulation, stability studies, we develop assays, analytical assays to measure the molecule in the presence and absence of other molecules. So related substances, impurities, stability, dosage form stability, solution stability, whether it is stable in powder form, how long it is stable. All these questions are asked, and then there are answers. And then also, what is not shown here is also synthesis, how well you synthesize the molecule, and what are the process impurities, and can we identify them, and can we reduce the impurities, etc. Also form a part of this preclinical development. Then if those things are positive, are encouraging, or very good results, then selection of candidate drug and preclinical studies is done. On one or two molecules that pass through all these filters would lead to preclinical studies. Now at this stage, in vitro drug toxicity screening becomes very important for selection of molecules. So any molecule that is going through lead to a preclinical development candidate selection stage, they all have to go through in vitro genotoxic assays and mutagenicity testing, and in vitro cytotoxicity assays. Why do we need this? It's because we want to identify that these molecules do not produce any genotoxicity. That means they are not detrimental to genetic material. They should not be disrupting DNA or any processes that are involved in DNA replication, protein synthesis, etc. Mutagenicity, they should not be introducing changes into the DNA that the DNA material become now mutated, and different forms of phenotypic expressions can be generated because of mutant genes. And it should not be in vitro cytotoxic. That means it should not kill cells. Now, having said that, here one trick I want to just deviate and tell you. Now, all these assays that are produce cytotoxicity or disruption of DNA or DNA toxicity or mutation, ability to generate mutations, these are red flags, and molecules depicting these activities are dropped. However, if you are looking at anti-cancer drugs, we are trying to pick up such activities because they lead to the efficacy or the expected therapeutic effect, because anti-cancer drugs should be killing cancer cells, so they should preferably cytotoxic. But selectively cytotoxic to tumor cells, but not to normal, regular human cells. So this is a differential cytotoxicity that would be looked at when you are developing anti-cancer drugs, but not here. Here, any cytotoxicity is a red flag and the molecule is dropped. And of course, um, the number of toxicity studies that needed to be performed before regulator gives permissions are based on the clinical experience of existing drugs. If the existing drugs have shown to produce certain toxicities and the regulator notices that those toxicities, and if many drugs are dropped because of these toxicities, then regulators would suggest that these toxicity assays be performed on the molecules that are being developed to ensure that such toxic molecules are not developed by mistake. Say, for example, we know that there are several drugs that are dropped from clinical practice because they produce cardiac toxicity by increasing arrhythmogenicity and introducing clinical arrhythmias and cardiotoxicity due to their involvement with a hERG potassium channel, conductance, and that hERG potassium channel conductance, prevention of that um, would cause QT interval prolongation that leads to arrhythmogenesis. So it is a regulatory test now that every molecule that is going for development, it should be tested on, hERG potassium channel conductance in CHO, that is Chinese hamster ovary cell line. Then there are toxicogenomics, which I will describe in shortly. This is also another predictor of toxicity. So why am I telling you all these things? I'll go back and then again tell you why. Because these are the measures that are introduced into during the development stage to eliminate toxicity and toxic material from getting approved in the clinic, such that safe and efficacious drugs are produced. So the what you are looking in pharmacovigilance is actually started much before a drug was born. But still, pharmacovigilance becomes a very important aspect. I'll tell you later why that is so. But you need to understand that drug molecules or candidate drug molecules under development go through rigorous screening to eliminate obvious toxicities. Elimination of obvious toxicities is referred to that you are ensuring that the molecule is safe. Okay, that is what is the drug safety is all about. Toxicogenomics is prediction of toxicity in a molecule by applying genomics principles. What that really means is that suppose if you prepare gene arrays from various tissues, gene arrays are nothing but, let's say, if it's a liver gene array, that means in all the hepatic genes that are expressed or put on a small chip. And the techniques of genomics and proteomics or genomics is in such a way that you can actually visualize which genes are activated and which genes are not activated by binding them with some oligonucleotides that can bind to activated genes. And these activated genes can be, whatever binds to them also has some fluorescent material. Therefore, in gene array, after it is expressed, all the genes that are expressed start giving different color signals. Okay. Now, you take normal hepatocytes and its genetic material on a particular, gene array experiment, and then look at which genes are silent, which genes are activated. Then suppose if you expose the same hepatocytes and another batch of those hepatocytes to a known hepatotoxin. Known hepatotoxins are like carbon tetrachloride, paracetamol at high concentration. These are all known to our rifampicin. There are so many drugs that are hepatotoxic, And suppose if the hepatocytes are exposed to them, and then the gene arrays are prepared, are exposed to gene arrays, then they will show which genes are activated by these hepatotoxic drugs. and most likely that those genes are activated, and an outcome of such activation is toxicity. Then you start comparing such activation patterns with other drugs that under, molecules that are under development, to see whether similar activation patterns are seen. It is like, thumb printing, let us say, that's a better word. For example, among certain, PPAR, PPAR gamma activators, thiazolidone dione drugs, which are used as anti-diabetic. The first drug that is introduced in the market is troglodyazone. But over a period of time, troglodyazone was dropped out of clinical development because it was found to produce hepatotoxicity in patients. Then subsequently, two other drugs were approved, rosiglitazone and pioglitazone. But the question that comes is that these rosiglitazone and pioglitazone are also hepatotoxic, or how can we establish that they are not? In toxicogenomics, in gene array assays, it's very clearly shown that troglodyazone actually activates certain genes, and those genes were not activated by rosiglitazone and pioglitazone. Thereby, a reasonable conclusion can be drawn that rosiglitazone and pioglitazone's gene activation pattern in hepatocytes is different from troglodyazone, and troglodyazone is hepatotoxic. But since these guys are producing a different pattern, these are not hepatotoxic. After reaching that conclusion, the drugs were approved in the clinic. Okay, and as expected, rosiglitazone and pioglitazone did not produce hepatotoxicity. Nevertheless, rosiglitazone produced some other toxicity, cardiac toxicity, and was withdrawn by the company itself. There's only one drug now available in the market, is pioglitazone. So such predictive assays will tell you, you have to put your molecules through those predictive assays to eliminate possible toxicities. Again, what is the intention of eliminating all those molecules? Just to ensure safety for clinical use. So what I'm trying to say, there are different steps during the development phase that are performed. Those experiments are performed to see if there is a red flag or not. So the red flag means, for molecule development, this experiment has proved a red flag, means that experiment, positive result in that experiment clearly indicates that this molecule has high incidence to produce that toxicity in the clinic. So you do not want to develop that any further. You don't want to take it to clinic and prove yourself correct. No. So you have to drop it at that stage and try to prevent clinical exposure to these toxic molecules of any clinical exposure. And drug development and preclinical phase, during preclinical phase, further evaluation of toxicity and safety is done for the selected molecules. Preclinical phase drug development the objective is to generate all the data that is necessary to file into an IND. It's as simple as that, And what is the data that you are going to file into IND? To demonstrate without any doubt that the molecule has activity and produce the required efficacy. Okay, so preclinical objectives are discover and develop patentable drug candidates and targets of relevance. That's what we are talking about. Generate data that gives reasonable estimate of clinical efficacy and safety. And also, because that's what is one particular objective. The other objective is that it is drug-like and it produces sufficient plasma concentration, and then you can predict a clinical dose and what sort of clinical plasma concentrations can be reached at the duration in a clinical trial. All these predictions are done during preclinical phase. The main objective is to provide enough evidence to obtain regulatory approval for first-in-human testing, because all this evidence is extrapolatory in nature, because it has not been tested in the human. All this testing is done in laboratory animals, or in cell lines, or on molecular biology assays, just to convince the regulator that this molecule is safe, it has efficacy, and it can get absorbed, and then a particular concentration can be reached. So please give permission to test in the human. At that time, it's better that the developer is not wrong, and then the better that the developer is absolutely correct. So what kind of clinical efficacy and safety experiments or estimates of clinical efficacy and safety experiments that you generate during preclinical phase? The preclinical development, IND enabling studies, first demonstrate efficacy and pharmacokinetics in animal models. The second, you perform an extensive safety pharmacology. You evaluate safety pharmacology of that molecule. Then you also do regulatory toxicology and toxicokinetics. Now here, we need to separate what is safety pharmacology and what is regulatory toxicology. I can tell you simply that regulatory toxicology is a study of understanding at what concentration the molecule is actually toxic. It actually starts to produce toxic effects on a laboratory animal. Okay. Toxicokinetics tells you what is the concentration and what is the exposure necessary to produce these toxic effects. And exposure in the sense that the blood levels and the extent of blood levels and the duration of their existence. In other words, when you generate the pharmacokinetic curves, the plasma concentration curves, the area under the curve is the time or the exposure for which the body sees this concentration. The extent of concentration for a duration. That is what we refer to as exposure. So in other words, when you administer high doses that are capable of producing certain toxic changes or changes that can be depicted as toxicity in various systems in an animal, at that level, what concentration and what is the exposure necessary? That would be, those are the outcomes that are obtained during regulatory toxicology and toxicokinetics studies. Now, when you perform efficacy experiments, you are monitoring a physiological effect which is modulated by the presence of this drug and which could lead to a therapeutic effect. That means I started with an example of antihypertensive. So let us think. The concentration that is necessary to decrease the blood pressure by a given amount of millimeters of mercury. Let's say blood pressure should be decreased by 10 to 15 millimeters of mercury. So the concentration that is necessary to produce a drop in blood pressure by 10 to 15 millimeters of mercury, that is the therapeutic effect. Okay. Why we said that's a therapeutic effect? Because that is the effect that is necessary to be demonstrated in the clinic. And what concentration is necessary and what is the pharmacokinetics of that. So that would also tell you the exposure to the body, the exposure necessary in the body to produce this therapeutic effect. That's the pharmacokinetics. Then 1,000 times higher concentration when administered produces ECG changes or deleterious changes on central nervous system. That's toxicity. What is the exposure necessary to produce that effect? There should be a great difference between these two. That means the pharmacokinetic exposure necessary to produce a therapeutic effect and a toxic exposure necessary to produce a toxicological effect. They should be very different. And this difference, if it is the larger, you call in very. This is how you have to establish that there is a great therapeutic index. Traditionally in pharmacology, therapeutic index calculations are done based on mortality, the dose that kills and the dose that doesn't kill and that produces the beneficial effect. But here we are not really looking at. In regulatory toxicology, we are going to find a preliminary range finding at what concentration actually the toxic effects can be seen, such that you can identify a concentration that is toxicity-free concentration, above which toxicity is seen. And we're talking about that concentration versus the concentration at which therapeutic effects can be seen. Now, that's the importance of regulatory toxicology and that importance of efficacy. Now, what is safety pharmacology? Now, let's again take the example of the hypertension. We are talking about an effect produced on blood pressure. The system affected is cardiovascular system, or maybe just simply blood vessels. That it causes an effect in the blood vessel diameter, so produces vasodilatation or relaxation and drops the blood pressure. That is the intended effect. It happens at a given concentration, let's say. But at that concentration or slightly higher concentration that the patient receives during repeated doses, or the other systems in the body affected. That means the central nervous system, the gastrointestinal system, the blood per se, the liver, the kidney, the muscle, and so many other systems, that the endocrinology, the pancreas, the blood sugar levels, are all they unaffected or affected? When the molecule is in the body at a given concentration sufficient to drop the blood pressure by about 10 to 15 millimeters of mercury, that's a therapeutic effect. At that level, is the molecule effective on all the other systems? Is it producing any effect or not? If it produces an effect, then it drops the blood pressure, simultaneously produces headache. It drops this, it increases blood pressure, blood sugar. All these things would come, and these are referred to as side effects. So safety pharmacology is ensuring that there are no simultaneous effects on other systems of physiology. That means what extent of side effects can be seen or no side effects. That evaluation is different because that evaluation is done at therapeutic concentrations, while regulatory toxicology is done at concentrations much, much, much higher than therapeutic concentrations. That's the difference between all the both or the three evaluations. So during preclinical development, you demonstrate efficacy and pharmacokinetics, you demonstrate the molecule is safety through safety pharmacology, and you demonstrate toxicokinetics through regulatory toxicology, and to show that the exposure, toxicokinetic exposure and pharmacokinetic exposure, are very, very different. So one certain more things about safety evaluation is safety pharmacology studies are provide dynamic and flexible approach of the potential function disturbance induced by new chemical entity. All the vital functions are evaluated, which is called core battery of tests. There's a whole list of them. The effect on CNS, and the effect on blood, and the effect on, gastrointestinal motility, respiratory, respiratory system, the pulmonary rate, depth of respiration, and so many things are done, which is again a huge subject. I'm not going to go through that, but when the molecule reaches the clinic, what is evaluated is that it is reasonably safe and efficacious and has toxicity, but at a very high concentration. Only then the molecule is approved for clinical studies. Okay. So when it enters into clinical study, successful IND leads into clinical trials. And there are, as I indicated to you, the clinical phases are phase 1, 2A, 2B, and 3. And during this, you are looking at proof of concept and safety. The safety is again a feature. And in clinical trials, for each clinical trial, you have these bodies are, established. DSMC, that is Data and Safety Monitoring Committee. Data Safety Monitoring Board, DSMC, and Data Monitoring Committee. What exactly these are is that the data is accumulated and data processed as per the protocols, and that when you are evaluating the data, there is sufficient safety or sufficient safety parameters are looked at. And if these bodies are not satisfied with the data that it is safe enough, the clinical trial can be stopped. Now, we simply understand that clinical phase 1 means it is tested in normal human volunteers, healthy human volunteers, in the dose ranges that needs to be tested in patients in phase 2A or 2B. And these people, these normal volunteers tolerate those doses properly. That's how we talk about. Successful clinical phase 1 means the doses are tolerated by healthy human people. But what's meant by tolerance? That there is no safety. That there is no toxicity and there is adequate safety. And the data that they collect on vital functions of those clinical phase volunteers did not show any untoward effect. That's very important. And that these people, plasma concentrations reached the expected levels. Because, when you administer a molecule to a patient and or to a human volunteer, healthy volunteer, and start looking at effects, probably if it's not absorbed, it will not produce any effect. So you have to ensure that first it's absorbed. So that means you have to tap the blood and look at the blood level. Suppose if the required levels are not reached, even then the phase is the clinical phase is not successful, because whatever you have predicted that at a given dose, these plasma concentrations will be reached. That objective is not met yet. So even then, the molecule fails in clinical phase 1. And these failures are attributed to either failure of pharmacokinetics or failure due to the molecule is not tolerated by the individual. That means it's not safe enough. So clinical phase 1 is a clear determinator of the molecule. If it is not safe, it's dropped. During clinical phase 1 and 2, actually you are collecting information from chronic toxicity studies in the animals, which are like 6 months toxicity or 1 year toxicity, and sometimes the carcinogenicity studies that you initiate during preclinical development, they run into a number of years. So as the data is accrued, that data is submitted to the DSMC during clinical phase trials, such that they try to combine the data that is obtained from clinical and the data coming from long-term preclinical toxicity studies to evaluate whether the molecule has sufficient safety to allow it to go to the next level. Okay. And next level is phase 2 is tested on patients. You are looking at whether the patients are producing the required response. So there is proof of concept that whatever you thought that by inhibiting this target, you would produce a given change that leads to a therapeutic outcome, has it reached? That was the proof of concept. And second, is it safe? So in phase 3, you would test it on larger number of patients in comparison to the current treatments or in comparison to placebo. That means patients receive a placebo which doesn't contain the active ingredient, but it's double blind. The patients or the people who has administered did not know which is the drug and which is the placebo, just to ensure that there is sufficient statistically measurable clinical outcome that is positive to approve the drug. The clinical outcome to reach the targets or objective, the milestones of the clinical trial. And the milestones have to be defined earlier. And simultaneously to look at if there is any toxicity, if there is any side effect, if there is any detrimental effect, such that at any stage, if a detrimental effect is seen, the clinical trial is stopped. Now, just to give you a current example, we're all now looking at, let's say, COVID vaccine trials. and which vaccine would come out first, because everybody is really interested in when the right vaccine would come. And there are at least some 145 or even more vaccine candidates under different stages of development. At least 9 vaccine candidates are in clinical phase 3, among which one vaccine is in India under development in phase 3. This is also referred to as Oxford University vaccine, which is being developed by Serum Institute of India. It is an intramuscular vaccine, one single dose, and it is an inactive, non-replicating viral vector vaccine, if you want to know. Globally, it is also being developed by AstraZeneca. During September, if you are following the headlines on all these things, the clinical trial of this vaccine, which is the most advanced, vaccine in clinical trials, the phase 3 has stopped all over the world, including in India. This is because one patient or two patients demonstrated certain muscular toxicity or muscular effects. Now they have to stop and evaluate the data to see whether that development of those conditions in those 2 or 3 clinical trial subjects is associated with the administration. That means there is a cause and effect, causal relationship between administration of this vaccine and that effect. So you have to pause, look at that and see whether is it because of the vaccine. Because it could be because of anything else that might have, coincidentally come out now that can be attributed to administration of the vaccine. See, here the biggest challenge here is that if there is an association between the vaccine and that effect, and if you fail to notice that or decided to overlook that and then approve the vaccine, there is a likelihood of several such cases would come when you start giving the vaccine all across the globe to all humans, right? But if you really not evaluate that, but jump to a conclusion saying that, oh yeah, that's because of the vaccine, so let's stop it. And suppose if that vaccine is not really responsible for the toxicity, you are depriving the population of an effective vaccine. See, so the decision making is very hard. So that's where the DSMC and DSMB, those committees would pour over the data and trying to establish the cause and effect relationship. And then, fortunately, after a week or so, the clinical trials were allowed to start again. So it is going ahead. Just to give you how extensively the safety is looked at during clinical development before approval of the drug. Okay. So regulatory submission for a NDA with all such data put together, if it is safe enough, then it is given a regulatory approval. And after that, post-marketing surveillance trials for clinical effectiveness evaluation, other indications, contraindications, etc. start, and pharmacovigilance starts. Now, see, in this lecture, almost like 45 minutes, I talked about the development phase only, because the drug discovery and pharmacovigilance, that word is used in the title. I want to emphasize to you that the discovery and development process for a drug, which takes almost on average 12 to 15 years, long process, the preclinical phase and clinical phase and final approval, would also is very, very seriously evaluated all possibilities of what can go wrong with the drug to ensure that it is safe. But the point is, you can only predict as much, but you can't really predict what happens when you start giving it to large number of populations. So that has increased the pharmacovigilance and the importance of pharmacovigilance. A few years ago, maybe 20, 30 years ago, the word pharmacovigilance is not seen, because when a drug is approved, it is believed to be, the regulator has decided, because there is sufficient data to say that it is safe and efficacious, and sufficient clinical evidence that it's producing beneficial effects and clinical outcomes. So therefore we approve. Then it is, released all across the globe, and then people start receiving it. More and more number of people receive it. More and more number of people receive it. But what is happening to them? Are all of them getting the benefits, or are there, untoward reactions, side effects, problems which go unnoticed? So here, the powerful statement is, medicines are supposed to save lives. Dying from a disease is something sometimes unavoidable. But dying from a medicine is unacceptable, because it is avoidable. You have introduced the medicine to treat a disease, but if because of that, and the patient has not died because of the disease, but because of the medicine that you introduced to treat the disease, is unacceptable. Okay. Then adverse drug reactions are among the top 10 causes of mortality in the US. If you really follow each drug very carefully, the clinical experience, and accumulate all the data, process the data, and look at what has happened to the patients that have received the drug, then all these facts come out. Unfortunately, in our country, I must say that drugs are given to much larger populations compared to anywhere in the world, because we have a huge population and we have a lot of drugs available currently. So many people receive the drug. Every day people are taking drugs. But if we don't monitor what is the outcome of taking all these drugs in those people very carefully, there are so many things that are unnoticed. So many side effects, so many adverse reactions, so much mortality that occurs because of the drugs goes unnoticed, because proper data is not collected and proper cause and effect relationship has not been established. It has been suggested that adverse drug reactions cause 5,700 deaths per year in UK. We're talking about thousands of deaths in that. The percent of hospital admissions due to drug-related events in some countries is about more than 10%. These are the evidences that indicate that drugs do cause, even though they are safe, do cause several problems. Okay. So pharmacovigilance is defined. Pharmacovigilance, are referred to as PV, is defined as the science and activities related to the detection, assessment, understanding, and prevention of adverse effects of any other drug-related problem. Adverse effects or any other drug-related problem. So it's a continuous monitoring of the drug usage in the clinic, and this monitoring is to detect, assess, understand, and prevent these adverse reactions. So pharmacovigilance is a very important and a huge field. It's not very small. So what is pharmacovigilance? If the question comes, pharmacovigilance is the assessment of safety of a marketed medicine. See, the definition is marketed medicine. So once it is in the market, once it is during clinical trials, you monitor all the patients that who have received the drug under close surveillance. That's why you call them as controlled clinical trial. But once the drug is approved and released into the market, there is no surveillance, there is no follow-up, because just think about, as a pharmacist, you are a pharmacist, dispenses a medicine. Somebody comes and then takes 100 tablets. The prescription says, use it for 3 months, so 100 tablets. And who is following what is happening to that patient after consuming those 100 tablets? This one is adherence to therapy, whether he is taking the 100 tablets or not. That is one evaluation, just to ensure whether the expected outcome is produced or not. Second is if he is adhering to the therapy and taking all the 100 tablets, one tablet a day for 3 months, in addition to the expected outcome, are there any adverse events? Are there any, side effects? Are there any toxic effects, produced in that patient for this, um, while using these 100 tablets for 3 months period? Now, as a pharmacy student, you can understand, would anybody actually monitor that? The patient himself is monitoring only if he is getting better. Okay. Very rarely he talks about some discomfort, Is it noticeable by him? Is it due to the drug? All these questions would come. So that evaluation is pharmacovigilance. Pharmacovigilance is to recognize and investigate unexpected clinical events that might occur once a new drug is in use. But a new drug, once it is in use, it becomes old drug. So in a way, pharmacovigilance is the monitoring of each and every drug in the clinic. The unexpected clinical events include rare adverse events that have low incidence, unforeseen interactions with coexisting clinical conditions or other drug therapies. Because an adverse event can arise out of anything. So we should be able to monitor, identify, and relate. This is very important. Now, let me define what is a side effect, what is the adverse drug event, and what is the adverse drug reaction. We use these sometimes, very synonymously, interchangeably. Side effect is the more common term. Any response to a drug, noxious and unintended, which occurs at doses normally used for prophylaxis, diagnosis, or therapy of a disease. We perform safety pharmacology to predict any side effect. Okay. I would simply say pharmacology has so many effects on the body. A drug can produce so many effects on the body, among which some are actually responsible for producing an efficacy, and we term them as therapeutic effect. The other effects which are simultaneously occurring on the other systems, if they can be noticed, then they form the side effects. Is the side effect tolerable? Is the side effect really, is preventing the therapeutic effect? In other words, is it a dose limiting side effect? That means you can't really administer any more because the side effect is more predominant. If a person receives a drug for lowering blood pressure or lowering blood glucose or whatever therapeutic effect, while producing that therapeutic effect, if the drug produces a very severe headache, unbearable headache, then it's a side effect which is dose limiting, and sometimes the person cannot take that drug anymore. So there are very obvious side effects which are usually detected during clinical trials. But then there are side effects which occur with low incidence, and those would be detected only during pharmacovigilance. An adverse event is an untoward medical occurrence that may present during treatment with a pharmaceutical product, but a causal relationship has not been established. In other words, I am taking a drug and then suddenly one day I experience a stomachache, right? A bit severe stomachache. So I can report that as an adverse event. But the stomachache could be because I ate something or due to an infection which is unrelated to the treatment. Or it could be that there is some change produced by the drug that caused, severe contracture of the stomach musculature, colic pain, and that is because of that. How do you know? First, that has to be observed, that has to be recorded, and that has to be investigated to see if there is a cause and effect relationship. So adverse events are like that. Adverse drug reaction is a response where a cause and effect relationship, a causal relationship has been established. In other words, every time you give the drug, that reaction occurs in certain populations. Then you know that it is an adverse reaction to the drug, like an allergy. I take the drug and my skin, there are rashes erupt all over my skin, but it doesn't occur in everybody. It only occurs in a few people. We call that those people are allergic to that drug. It's an adverse drug reaction. So there should be a caution to that. But in the clinical practice, how do you identify all these? That is the subject, or the science of doing that is pharmacovigilance. Then a golden question arises, is that is pharmacovigilance needed when drugs are approved after establishing safety? This question, has been there in every. If I say that this is a side effect, but then it has undergone clinical trial, why it was not stopped during the clinical trial, or why it was not detected? There are examples. There's explanations why it is not. Clinical trials aim to establish safety of a new medicine using a smaller population of patients, very well-controlled population, controlled number of doses given for a controlled period, and they want to see whether there are any side effects, any adverse events occur in those patients. These trials detect harm due to most common adverse events, those adverse events that are very common, that occur in most of the population and with high frequency. That's what is observed. While pharmacovigilance is the evaluation of safety in less selected populations over longer periods to obtain a comprehensive evaluation of safety in routine care throughout a drug's life cycle. I can give you an example. If we say that there is a incidence of 0.1%, of developing a headache, what that means is if 1,000 people receive the drug, one person would develop a side effect of headache. So the incidence is 0.01%, or 0.1%, whatever. 1% is 1 in 100, 0.1% is 1 in 1,000. Okay. If it is in 10,000, then you say 0.01%. So if there is a side effect, has an incidence of 0.01%, and you test them in 1,000 patients, what is the likelihood of that side effect showing up in these 1,000 patients? Because it occurs 1 in 10,000, so it has very low probability. But if you monitor these drug usage in 100,000 patients, almost 10 to 12 of them cases would spring up. Okay. And if it is in a million, then almost 1,000 cases would show up, or 100 cases would show up. That becomes more obvious. But you have to monitor million patients, and that cannot happen during a clinical trial. You cannot have a clinical trial where you enroll million patients and run for 10 years, just to see whether there is a incidence of 0.01% side effect. Then the drug would never get approved. It can never be released into the clinic, So therefore, the evaluation of safety during clinical trials usually establishes safety, in smaller populations, and the most common adverse reactions are seen. But pharmacovigilance would identify not so obvious but low incidence toxicities. So pharmacovigilance with drug safety during trials, the differences are pharmacovigilance is a continuous evaluation of safety of a drug throughout the life cycle. Clinical trial, drug data monitoring committees, objective is to prove safety for obtaining approval. That there is no obvious toxicities. It is quite safe. That's what their conclusion is. Pharmacovigilance aims at identifying all adverse events, common as well as rare. It continuously monitors patients to see what sort of side effects or adverse events that occur during the life cycle. In other words, here there is an example. I have received the drug several times, and every time I have no problem. But the 10th time or 15th time I started taking, I developed a severe allergy. It is likely because the first exposure, second exposure, third exposure to the drug might not have generated a allergic response in my body. But over a period of time, there is a change that occurred. I suddenly became allergic. So we cannot say that you don't have drug allergy because the first, second, and third, or first five exposures to the drug did not produce that. It doesn't mean that over a period of 100 exposures you would not produce that. If you can recall cases when people who ate wheat or who ate peanuts all their life has no allergy, but suddenly over a period of time they suddenly develop allergy to peanuts or gluten allergy. What has happened? Their constitution has changed that now it cannot tolerate gluten anymore, so they became allergic to all wheat products. Doesn't mean that they're born with that allergy, although there are some people who are born with allergy. So please remember, the individual constitutional change would also make them respond adversely to a drug. But it doesn't mean that because they have changed and the drug can produce that effect and it is their fate. No, we have to follow that to see what is the probability of such cases occurring. So that's pharmacovigilance. It's a lifelong safety evaluation of a drug. The clinical trial, data monitoring committee are only concerned about identify the most common adverse reactions. So drug safety during trials and pharmacovigilance are different. Pharmacovigilance starts after the clinical trials and once the drug is established to be safe for clinical use. But you cannot, ascertain that it is forever safe. That's where the pharmacovigilance comes into picture. Here is a graphical explanation of, what kind of side effects or what kind of, how patients would respond adversely to a drug. Now, the entire square here, which has, let's say, 5-6 bright spots, the entire square represents a huge population, and the huge population is receiving the drug, uniformly, and only 6 people respond with this side effect. But during clinical trial, a smaller population, when they are exposed to the drug, that population may not include these 6 people. So in the clinical trial, we put it on the boundary that it might even not noticed during a clinical trial. So these are considered as low incidence side effects or adverse events, but that can occur after prolonged use of drug or when the drug is given to larger number of people. Because during a clinical trial, it may be up to 1,000 or a few thousands of people are exposed and they are all controlled. That means there are exclusion criteria, inclusion criteria, their diet is monitored, everything is monitored. But in the clinical practice, when the drug is approved for use, there is no monitoring, there is no control on the patients. It is given to all eligible patients all across the globe. There are genetic differences among them, there are racial differences among them, there are environmental differences among them, their food habits are different, and comorbidities and concurrent use of other drugs, nothing is controlled there. So there is a higher chance of such effects can be seen. So that's why pharmacovigilance continues. Pharmacovigilance becomes important. So what are the factors influencing adverse drug events? A typical onset of events such as allergic reactions. I already gave you an example of allergy can arise any given time. Disappearance of events after cessation and therapy and recurrence with restarting the therapy. So how do you, what you call associate? Now, in another slide, actually, what is to be done in pharmacovigilance is given. This is pharmacovigilance methodology. Broadly, a 5-step process. Collect information. First, you have to collect the information. How do you collect the information? By observing, through passive or active surveillance. Either they report to somebody and then the report is, is recorded somewhere. or you come to hear that somebody has received a drug and that person has responded, or you directly monitor that individual to see, oh, you have taken this and what happened, so that's mandatory report. Identify the incidents or ADRs or ADEs. Now, I have defined ADRs which are with a established link, cause and effect relationship to the drug. ADE is occurring because of the drug or not because of the drug. It's a simultaneous, a coincidental occurrence, and you have to study to establish that link. Evaluate ADRs through de-challenge and re-challenge testing. That's exactly what the previous slide was trying to show. Disappearance of events after cessation and recurrence after restarting the therapy. So one way of identifying that is, in a patient, if you identify an ADR, then you stop the drug and it should disappear, and then if you restart the drug, it should reappear. That's a relationship. And most important is, the observation of pharmacovigilance has to be followed by ACT, by reporting and to monitoring centers and regulators. The data has to be reported. The data has to be collated. The data has to be made available in data banks. Why I am saying that is, a particular drug is being used in India, in Japan, in South Africa, and maybe in Brazil, let's say. And a particular reaction has been reported in Japan and Brazil, but we don't know about it, and we start giving the drug here and then start observing the same reaction and trying to establish a cause and effect relationship, whether it is due to the drug or not. But if you can access the data, pharmacovigilance data from other countries, and if it's already reported elsewhere that administration of this drug has caused అంటే నాకు you are in the show. Hello, can you see? Hello? Hello, can you hear me?
Yes, sir, we can hear you and see the slides, sir.
Oh, I just heard you. Okay, that's fine.
We can hear you and see the slides, sir.
Right. So I have to go. So now, can you see the slides in full screen?
Yeah.
Yeah, okay. I'm sorry, technical hitch. Suddenly
Yeah,
blanked out. So
yeah,
we're
yeah,
talking about act by reporting. So what I'm trying to say is, you have to collect information, identify the incidents as ADR or ADEs, evaluate through de-challenge and re-challenge, which is, stop the treatment to see if the ADR disappears, and re-challenge to see if it reappears. And most important is then this needs to be reported and collated with information that might have come from different countries on the same type of effects. Which was, I was telling an example that if suppose in a global use, if there is such cases reported in Japan, Brazil, South Africa, but if you and India, and if we do not have access to that data, then you do not know whether such effects have already been observed. Then you try to establish cause and effect relationship, whether there is a relationship between the drug and these adverse event. But if you can access that data, then you already know that such cases have been reported, so therefore it is more likely because of this drug. Then continue to monitor for such reactions, because now it would come under the uh, various possible adverse reactions or adverse events that this drug can cause. And so what to look for, because it's always that if it's a known side effect, you look for that. But if it's an unknown, you have to observe and associate and add to the list. This is a 5-step process of pharmacovigilance. For that, there are methodologies to study, you know and spontaneous reporting systems, such as documenting and reporting of adverse events that occur when a new medicine is used in clinical practice. Most regulatory authorities operate a database for storing and analyzing adverse event reports. So you would report into the pharmacovigilance centers. Active surveillance is identification of potential safety problems through regular and systematic collection of clinical information using a specified population of patients prescribed a new drug under normal clinical conditions of use. Now, how do we do that? Is again, it's an extended clinical trial. So, once if you are involved in a, a pharmacovigilance program of a new drug. Now, I've been talking, or I have written somewhere that when the COVID vaccines, they started to be distributed. They're available. They are made available, and we start immunizing populations. Two things that need to come is one, continuous monitoring of efficacy, whether they're effective. Because you are making an injection of an antigen, and whether the desired response is, that means development of antibodies. You have to monitor that for some time. But at the same time, adverse events. What happens to the patients who receive these vaccines? So the pharmacovigilance starts. And since this vaccine, let us say, is being given all over the country, so there is, if you start collecting data from every single individual, then you require a large pharmacovigilance workforce, and there is so much of data that is collected because it inundates the entire system. So therefore, then we follow, designated populations and say that in a given area, 100 people or 200 people who receive this vaccine would be monitored for a month or two. But such given areas are not just one, maybe 1,000 or 2,000 areas in the country, and the data is collated from all those areas to see what kind of safety profile the vaccine would have. And depending upon the you know detection and association of adverse events, you would start increasing the size of the study, and probably number of pharmacovigilance investigators involved. This is how active surveillance can be developed. But we must also have a very good reporting system. Where do you report that? Suppose today you have observed something, and where do you report that? You should have an idea of that also. So patient registers or registries. Registers comprising of data collected on a defined patient population for a defined period of time. Drug registers, condition-specific registers. All these data has to be fed into that. The aim is to increase safety knowledge of the product in long-term use. As I said, when you observe something and associate and convert an ADE, adverse event to a ADR, it should be added to already known ADRs with that particular drug, and that should be put in a drug register, drug information register, that when people start looking at the drug information register, they know that, oh, okay, this drug or this vaccine would now known to produce even this effect. This is how information is registered and gets into medical records. Records from medical practices, community pharmacies help in conducting epidemiological studies on drug safety. This is another thing, when your pharmacovigilance and efficacy studies accumulate data and put it in medical records, then epidemiology starts. Pharmacoepidemiology is evaluating efficacy of a drug in populations, and it also compares how effective that drug in different populations under different conditions, and whether this population should be given that drug or not based on this efficacy. So in a vaccine, then they would also evaluate the take rate, how many people have actually taken and responded by producing good amount of antibody titers, and what is the effectiveness in that population. Has the prevalence of the infection decreased after the vaccine is given? Now, but the prevalence of disease has not decreased, but the prevalence or the incidence of side effects increased. If it comes from pharmacovigilance, then the vaccine is totally ineffective. All these studies, all these data together, whether pharmacovigilance or phase 4 post-marketing surveillance, which is efficacy monitoring, or pharmacoepidemiology, all these studies are designed to provide better data and a complete understanding of how the drug, when it enters the market, is actually behaving, or how the populations are responding to this new drug. This is very, very important aspects. So pharmacovigilance in India, just a history, is that it first started in 1989. DCGI started ADR monitoring at Delhi, Mumbai, Calcutta, Lucknow, Chandigarh, and Pondicherry, for spontaneous reporting, intensive hospital monitoring, and focused reporting. Subsequently, India joined the WHO monitoring program in 1998. National Pharmacovigilance Center started at All India Institute of Medical Sciences in Delhi, and WHO Special Center started at GS Medical College, KEM Hospital, Mumbai. These are all historical facts, so I'm just reading out. And second attempt in 2004, World Bank aided pharmacovigilance program, the National Pharmacovigilance Center, and the program PVPI, Pharmacovigilance India, and Pharmacovigilance Advisory Committee formed. And there are two zones formed: Northeast at AMIS, All India Institute of Medical Science, and Southwest at GS Medical College, KEM Hospital, Mumbai. Now, 4 regional centers: North, East, South, and West, and peripheral centers. Mumbai center was sending ADRs to World Health. You have to have regional centers to accumulate the data, and this is connected to a central data repository or data bank, and that has to be connected to a global bank. So this process is very very intricate, but very methodical. Ongoing pharmacovigilance program of India started in July 2010 under CDSCO, Ministry of Health, Family Welfare, with National Coordination Center at All India Institute of Medical Science. Subsequently, the national center shifted to Indian Pharmacopoeia Commission in Ghaziabad, and currently the former DCGI, Dr. G.N. Singh, is in charge of that. Even while as a DCGI, he was still the member secretary of the pharmacovigilance program. In one of the launch programs I attended, and There are pharmacovigilance courses offered where the 5-step process of how you collect information, how you identify, how you evaluate, how to act, and how you monitor and how you report can be taught, because it is just not a program where you can observe easily, because there's a very method, methodical approach one should have, and an understanding of pharmacovigilance. And in a nutshell, pharmacovigilance is continued monitoring of safety and effectiveness, because safety has a impact on effectiveness. If it is no longer safe, then you're not really looking at how efficacious the drug is, because it's not safe to use, so you have to discontinue. Through pharmacovigilance, you can identify those severe drug interactions that could cause mortality. So therefore, pharmacovigilance is essential for reducing mortality and morbidity that a drug can produce on prolonged use in larger groups of population. So in a nutshell, this is drug discovery and pharmacovigilance. I thank you all for giving me this opportunity, and I took a lot of time because Madam said that as long as you can take, but I have a tendency to get carried away because I want to give all information that I have. Hopefully all the people who listen to it have now a fairly good understanding of what this is all about, at least some background understanding that if you listen to more structured talks, then you can relate better. Thank you very much. All the best. Over to you.
Thank you very much, sir. Thanks a lot for your wonderful talk. We are very fortunate to have you over here today in this guest lecture. There are few questions, sir.
Yes.
The first question was, is it possible, how can the students can select projects in pharmacovigilance?
If you really want to do pharmacovigilance as a part of an undergraduate or a master's program, probably you need a bit of formal training, a course or something to run into how exactly you identify and associate, etc. Then the second is by simply choosing. If very easy to choose are a new vaccine introduced. I keep on giving the vaccine example. Why I keep on giving the vaccine example is very simple. Because once I had a very interesting conversation with Dr. Krishna Ella, the chairman MD of Bharat Biotech, and he is in the forefront because he has the vaccine Covaxin, which is in phase 2 and about to enter into phase 3. It's an intramuscular inactive viral vaccine. Some few years ago, he was talking about, because he has they have introduced several vaccines, the rotaviral vaccines, and now I can't really tell you how many vaccines they have, but the combined vaccines, combination vaccines, etc. Then he asked me a question saying that can I get PharmD graduates in a given region? Say I was at that time with St. Peter's and in Warangal, and so he specifically called me and asked me, can you give me about 8 or 9 people who are graduating, just because we are releasing the drug, the vaccine in Warangal region. So I need people to monitor pharmacovigilance. Okay. And the required training, when they join the job, they would provide the required training in the company. But that suddenly hit me, like, for every new drug, for every new vaccine, you require pharmacovigilance. And with a formal training of how you monitor, how you identify, how to associate, how you associate, you can choose any drug and start looking at a given population in a given area. those people who are receiving this drug, maybe you can start from a clinic or you can start from a pharmacy and start following those people about what kind of discomforts or events or something that they can report. Okay, in a undergraduate graduate program. That's the best I can give answer.
Okay, sir. And that means, is it possible to do the projects without going to attending the hospitals and clinics, sir? And based upon the review of
Uh,
literature, shall we plan? Is it
no, you
possible?
don't have to go to a hospital, but you have to have access to information of patients who have received a given treatment.
That means literature.
No, literature would tell you what.
Okay.
Basically,
How to
literature.
consult the patients.
Okay, I'll tell you. Suppose you have read about pharmacovigilance of a particular drug, and there is something that is reported in that. But that literature, that study was conducted in, let us say, Bihar, or let us say in China.
Yeah.
Because the study can be from anywhere. Now, based on that, you want to start a study in your own region, you can very well do. Because if those people, if the reported literature has already indicated that there are certain adverse events that they have identified, you can predominantly look for those here. Okay.
Hmm.
But
Okay.
at the same time, people should not miss others, because in pharmacovigilance, that is the most important aspect. You can't leave, you have to identify, monitor, identify, and associate. If you
Oh,
only look for something that is already associated for, you are only indicating what is the incidence of that side effect. But if any other new effect,
okay.
you know, any other new event, if that occurs, then you would not have that. That is what my point is. So yes,
Okay.
you are right, Doctor. That you can take the help of published literature to start a project or program. But
Okay.
let us say drug A is received, but how do you know who are taking that drug? Either you have to
Yeah.
go into a pharmacy and try to find out the information, or a clinic,
Yeah.
to get that information.
During this COVID situation, I think it is not possible to go to clinics or hospital. If the student wants to do the project, then what is the suggestions from you?
Because this is not a hypothetical subject that they can predict.
Yeah.
They can do a survey of pharmacovigilance of certain drugs and then make a report on, collate a report, such that report would be more useful because of studying all those. It's like a review article. One can do
Yeah.
that.
Yeah. Hmm.
But that is preparing a review article on pharmacovigilance of a particular drug. I'm alluding to a particular effect already. I mentioned, because when the COVID vaccine hits the scene, everybody receives it, right? Now, when they receive it, we do not know. But
Hmm.
all the people in your college would also receive, all the faculty, all the staff, all the students. That is what is meant for. The vaccination to protect population from coronavirus infection needs to be given to each and every fellow. There may be a priority that some people would receive first and some people would receive later. But then pharmacovigilance becomes such a humongous task of doing that. So I'm
Yeah.
trying to say, like that, you can choose. Maybe you can choose some vaccines, children's vaccines, or if there is a new vaccine, a flu vaccine. What is it? To understand the pharmacovigilance of that vaccine, the population is given, or any drug. But what else we can? What will be the criteria for natural product assessing in pharmacovigilance? Sir? It's a wonderful question.
Yeah.
That question just popped up in the screen. So you want me to answer that?
Yeah.
Yeah.
There is one more question,
Please.
sir.
No,
Yeah.
you sorry reading. Yeah.
No, that's okay. If if you want to prioritize, then ask me other questions, then I'll come back to this question. Go ahead, madam. Ask me the question.
Okay, sir. Thank you. What will be the criteria for natural products assessing in pharmacovigilance?
Oh, that's what I was saying. That's the
Same.
Same thing.
you
Is
know, any natural product literature, when I look at that, you know, there's obviously one statement is made, that is natural products are safe, and people are now going more and more into natural products because the synthetic drugs are toxic. Okay, it is a sweeping statement that, do we really know whether all the natural products people are taking are safe, that they are free of any side effect or free of any adverse event? You know, I don't know. First, we have to identify which is the natural product that is very often taken by populations in order to do pharmacovigilance. You know, how many of you take? Because I can mention, I haven't taken any natural product or herbal extract or any preparation over a long period of time. So you can identify, you know, suppose let's say Chyawanprash. How effective Chyawanprash is, pharmacoepidemiology, what is the benefit of populations taking Chyawanprash, and what could be the side effects or adverse events that could be associated with Chyawanprash use? That can be the pharmacovigilance study. Okay. But is there a dose? Is there a regular regimen that they follow? Otherwise, how do you define your pharmacovigilance? This is my question. If there is a bottle of Chyawanprash at home and then I take it once in a while, a spoonful of it, then how do you monitor people like that? So there may be difficulties in doing that. But if people are following a anti-diabetic medication or some CNS medication on a herbal based, then one can follow the same principles of pharmacovigilance. Because pharmacovigilance is not looking for the obvious. Pharmacovigilance is looking for something that might occur, right? Which are unpredictable side effect. So the same criteria can be used, you know, detect, associate, act. But here the drug, in the place of the drug, you write a natural product. You have to define the natural product though. That's what the point is. I hope I asked.
Yes.
Yeah.
There is one more question, sir. What are the softwares available for pharmacovigilance?
I don't know the question. I don't know the answer. So, is that the question related to how we access the reporting system and report?
Yeah, it is there, sir. It is there, some like databases.
Yeah, I think you can
She is asking
contact.
that one. Yeah.
You can contact pharmacovigilance PVPI program Ghaziabad to get an answer. I don't know what it's like.
Yeah.
For
Correct.
any drug related query, drug related information, or for any thing you want to monitor with CDSCO, there is a portal called DAVA, you know, DAVA, you know, DAVA, medicine, you know. Like that, there can be a portal, but at the moment, I do not know. I know the guy who is looking after that, you know, what is that, his name? But name skip. He also appeared on several platforms and gave lectures. You know, I'll try to recollect. Who works under Dr. G.N. Singh? You know, somehow his name is skipping my attention.
Yeah, it's true, sir.
Yeah. Maybe I can get that
One-
information from him. So,
yeah. So one more question.
is it mandatory learning pharmacovigilance course for 6 months to get into field?
Get into the field.
Is it mandatory to learn driving before start driving a car?
Yeah.
I have mentioned that there are
5
programs
steps.
available, courses available to be more competent and confident to perform that. One needs to take that program. Without that, it is very difficult because what my lecture only indicates the information that I have and collating that information with what I already know, and it's highly hypothetical. But if you ask me, can you lead a program, pharmacovigilance program out on the road? I probably need to have some training of how to start and how to progress with that, right?
Yeah.
That is absolutely essential. Even for me, I'm not excluding myself from that. If I want to get into, if there is a structured program, I would like to take that program. It is mandatory, it is necessary. It's just like you have to learn driving to
Train.
drive a car.
Okay, sir. Pharmacy students, they are interested to enter into pharmacovigilance field. Some of the students, so what that is their doubt, whether we have to learn something, some training course before entering into pharmacovigilance. Otherwise, we can get directly by
How
this
is it possible
thing.
without knowing
One can start directly. It's not that
logical?
it cannot, but one
Yeah.
has to read a lot of literature, understand the methodology, because we talked about, when we say efficacy of a drug, you know, now how do you determine efficacy of a drug? It is called outcomes research. And there is a structured way outcomes research has to be learned in order to detect the outcome of a treatment in a patient. So there are scales and there are quality of life measures, etc. It's easy to talk, but if you ask, you are so much talking about outcomes research, and you're talking about outcome, therapeutic outcome, positive outcome, and this. Can you do that? I would simply say no. I need training, because saying something, trying to understand something is different from actually doing it. For that, you require training. Then you can do it better. We manage projects, no problem. But when you want to be more effective in project management in industry, then you have to take project management courses. Is it mandatory? It is mandatory for you to become an effective and efficient project manager.
Yes. Thank you, sir. Thank you for addressing all the questions raised by the audience. Thanks a lot for accepting our invitation and spending your time with us. We are very fortunate to have you here today. Thanks a lot, sir.
I thought I would be more fortunate to be physically present, because I came to your college on a couple of occasions. I liked it.
Yeah, yeah.
So I
Okay.
tell everybody that I'm looking forward to that occasion when I can physically be present there, because then I can enjoy the ambience. Because your college, particularly in that area, Barkatpura area, that I like
Yeah,
that area very much.
yeah. Thank you so much, sir. Your patience almost 2 hours, more than 2 hours.
No, that's my pleasure. If I can add value by
Yeah,
spending 2 hours, it doesn't matter. But I hope I
yeah.
haven't bored the students. That's what is most
Yeah.
important for me.
No, 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 no boring, sir. It's in detail, in detail. It was.
Yeah.
Now we got the clarity on that. It's not only adverse reactions or adverse effects. So from the step one in the drug discovery, it is involving. Yeah, that
Yeah,
is.
exactly. Because certain
Yeah,
side effects are already known. So pharmacovigilance is not monitoring how frequently or how intensely those same side effects would appear after the drug. No, that's not this thing.
correct.
Are there
Yeah,
any
yeah,
new ones that are
yeah,
associated with the drug use? So it has to be observed, and then a cause and effect relationship has to be established. So that is where structured methodology is necessary, and that
yeah.
for that structured methodology you need to undergo training.
So it is nothing but safety monitor. Pharmacovigilance is nothing but the safety monitor. That's all.
And
Whatever the
continuous,
safety it may be related.
yes, exactly, continuous lifelong safety monitoring of a drug.
Yeah,
But then what are you monitoring? What are you looking for? That's what it is. So there can be, if you are looking for very obvious things, you may miss the unobvious things.
yeah,
So it is, research is always exploring something that is not found yet.
yeah,
Research is not exploring to find what everybody else found again, you know.
Yeah.
So that means you have to have an eye for the unobvious, you know.
Yeah.
Here also,
Say. Correct.
it's easier said, you know, but actually when you are looking, the difficulty comes.
Yeah, yeah. It's true, sir. Thank
That's
you.
why structured training is necessary.
Yeah, correct.
Kalai Selvan, his name is, I
Oh,
suddenly remembered, you know, E. Kalai Selvan. You know, maybe you
oh,
can invite him to give a talk, you know,
okay, sir.
because now it is online, so it's very easy. And
Yeah, yeah.
Kalai Selvan can give you. I was kicking myself. You know, I know this guy so well, but how come I don't remember? Have you come across him? If I have his number or you know, email ID, I will send you.
Okay, sir. Thank you so much, sir.
Right?
We'll
Yeah.
arrange one day. Thank you,
Yes.
sir.
Then
Okay,
he
sir.
can tell you how the data can be entered and where is it available, what kind of interfaces you need, and what if there are any structured programs. Okay, you
Okay, sir.
remember? I'll tell you one small incident. You know, once in IPC we conduct a um, national event called you know, National Electrocution Competition. In
Okay.
one semi-final or final, when the students are presenting, that time the talk was about pharmacist and drug safety or something.
Okay.
Uh, Suddenly, Dr. G.N. Singh sent a message to be announced during that talk. I mean that event at the end. You know, I
Okay.
think Dr. Ramkishan represented, and then he gave. He was so impressed that he said that all these people, or the you know, graduate B Farm, M Farms, etc., they would be interested in taking them in the pharmacovigilance program, you know,
Yeah.
particularly. But announcement was made during that event.
Okay.
I
Okay.
recall very well, you know. So we
Sir,
should
this
use, yeah.
Eliva cell, is it from Tamil Nadu?
Kalai Selvan, yes, but he
Kalai.
works in Ghaziabad.
Okay, sir.
He
If
is the
possible,
principal.
yeah.
He is the principal investigator or principal scientist of Pharmacovigilance Program of India. But this was
Okay.
something 3-4 years ago, so he might have changed it now. I do not know. I know, but he's
Okay.
still in the
Okay.
program. I know. I
Okay.
might have his email ID or something. You know,
Yeah, if possible, please help us, sir. We'll conduct one day. Why it needs for the students.
correct.
Yeah.
I can do that. Yeah,
Okay, sir. Okay, sir. Thank you, sir.
it's my pleasure.
Yeah. Thank you. Thank you so much.
Right.
Okay.
Take care.
Bye,
All the
sir.
best, and I know all of you. Please stay safe. You know,
Yeah. Definitely, sir. Thank you.
so being
Thank
safe
you.
is utmost priority now.
Yeah.
Okay.
Yeah.
Right.
Yeah. Yeah. Definitely, sir. Thank you. Thank you.
Thank you, sir.
Thank you.
Thanks a lot.
Bye.
Bye.
Thank you, sir.